A revolution in science. No sperm, no eggs, yet life begins. Scientists have achieved the unthinkable, creating synthetic embryos in the lab. This breakthrough challenges everything we know about reproduction and raises profound ethical and moral questions. How did they do it? What could this mean for the future of medicine? And are we ready for the implications of this scientific leap? So watch the full video and don't forget to hit the like button and subscribe to our channel for more videos like this one. A revolutionary discovery. The world of reproductive science has just taken a monumental leap forward. What if scientists could create embryos without sperm or eggs? This groundbreaking breakthrough could change our understanding of human development, medicine, and even reproductive technology. The latest research in synthetic biology has revealed a bold new frontier, creating human-like embryos using stem cells, bypassing the need for traditional sperm and egg cells. But how does this revolutionary technique work, and what are the potential implications? The science behind synthetic embryos, a leap in stem cell technology. The experiment begins with stem cells, the remarkable building blocks of life. Scientists have been experimenting with human stem cells for years, with some derived from donated early embryos and others from induced pluripotent stem (iPS) cells, which are reprogrammed from skin cells back to an embryonic state. These stem cells have the potential to turn into any type of cell in the body, making them invaluable tools in scientific research. In this latest breakthrough, scientists have taken human stem cells and placed them in a specialized medium that pushes them to their most primitive, or naive, stage. This allows the cells to begin developing in a way that mimics the early stages of embryonic development. After carefully orchestrating the growth process, the scientists grouped these cells into aggregates of 120 cells each, and what happened next was nothing short of extraordinary. Self-differentiation. Cells taking matters into their own hands. In the experiment, roughly 1% of the aggregates began to differentiate. They started sorting themselves into the correct structures, mimicking the earliest stages of embryonic development. This self-organizing behavior is one of the key breakthroughs of the experiment. While traditional embryonic development relies on sperm and egg fusion, this new method allows scientists to create a sort of artificial embryo that begins to follow its own developmental path, guided by the natural properties of stem cells. Although this process can only progress as far as day 14 of embryo development, it provides scientists with a unique opportunity to study embryonic development in ways that were previously impossible. The problem with traditional methods has always been the ethical and logistical limitations of accessing and studying human embryos. With this synthetic embryo model, researchers can get a closer look at what happens during the first few weeks of development. A black box. Understanding organ formation. One of the biggest mysteries in biology is how human organs form in the early stages of pregnancy. By the 5th week of pregnancy, a human embryo has developed the basic structure for all major organs. However, the exact process of organogenesis, the development of organs from stem cells, has largely remained a black box, as it's nearly impossible to study directly. This synthetic embryo model is aiming to shed light on this process. With engineered stem cells, scientists can now manipulate genes and hormones to better understand how and why certain organs develop. By tweaking these genetic components, researchers hope to identify which genes and hormones are crucial for organ formation, paving the way for future advancements in stem cell therapy and regenerative medicine. The goal: a model for human embryonic development. The long-term goal of this research is not to create a fully functional human embryo outside the womb, but rather to create a model for studying embryonic development. By understanding the processes at play, scientists hope to improve their ability to generate specific types of cells that can be used for research, drug testing, and even potential future therapies. Though it may be tempting to imagine this discovery leading to the creation of synthetic babies or artificial pregnancies, that is far from the reality. The research makes it clear that the environment inside the uterus is vital for the proper development of a human embryo, and replicating it outside the body is currently impossible. For now, the focus is on advancing our understanding of early human development and improving medical applications. Potential applications beyond the lab. So, what does this breakthrough mean for the future? There are numerous potential applications for this technology. For one, synthetic embryos could provide a valuable resource for generating cells that could be used in regenerative medicine. For example, cells from these synthetic embryos could be used to treat conditions such as heart disease, diabetes, or neurological disorders by replacing damaged tissues with healthy lab-grown cells. Furthermore, synthetic embryos could serve as models for drug testing, helping researchers determine how new medications might affect human development in the earliest stages. By studying how different compounds interact with these synthetic embryos, scientists could identify potential risks and benefits before drugs are tested on actual human embryos or fetuses. As with any major scientific breakthrough, there are significant ethical concerns surrounding the creation of synthetic embryos. While the ability to create these models could revolutionize medicine, it also raises important questions about the definition of life, human rights, and the potential for misuse of the technology. For instance, if synthetic embryos can be created and studied, could they ever be used to develop cloning technologies or experiments that blur the line between human and artificial life? Ethical guidelines will need to be developed to ensure that this technology is used responsibly and for the benefit of humanity. The focus should remain on medical advancements and the improvement of human health, rather than exploring dangerous or morally questionable uses. Challenges ahead: refining the process. While the creation of synthetic embryos represents a monumental achievement, the process is far from perfect. The model currently only works up to day 14 of embryo development, and there is still much to learn about the complex journey from stem cells to fully formed organs. The challenge will be refining the protocols to create more accurate and stable embryo models, allowing scientists to study longer periods of development and gain deeper insights into the mechanisms at play. Furthermore, creating a truly identical human embryo remains an elusive goal. The womb plays an essential role in the growth and development of a human embryo, and it is unlikely that synthetic models will ever fully replicate this process. However, the knowledge gained from these experiments will have profound implications for regenerative medicine, gene therapy, and our understanding of human biology. A new era in embryonic research. The creation of synthetic embryos without sperm or eggs is a remarkable step forward in the field of developmental biology. While we are still far from creating fully developed human embryos outside the body, this discovery opens up new possibilities for studying human development and advancing medical science. By refining this technology, scientists could one day harness the power of synthetic embryos to treat diseases, repair damaged tissues, and improve the quality of life for millions of people. The road ahead is both exciting and challenging, but the potential for breakthroughs in stem cell research, regenerative medicine, and developmental biology is vast. As science continues to push the boundaries of what is possible, one thing is clear: the future of medicine and human biology is being reshaped before our very eyes. What do you think about the ability to create synthetic embryos without sperm or eggs? Is this the future of science or a step too far? Share your thoughts in the comments below. Don't forget to like, share, and subscribe to our channel for more cutting-edge discoveries and debates. Science is pushing boundaries like never before, and we're here to keep you informed. Thanks for watching. Stay curious, stay thoughtful, and we'll see you in the next video.